Hello, everyone. My name is Donna Cornwell.
And my name is Kelly Seacat, and we're the hosts of Scratch Space.
Scratch Space is a virtual forum hosted by the Lucas Artist Residency Program at Montalvo Art Center, which is located in Saratoga, California, on the ancestral lands of the Ohlone people. And with Scratch Space, we're bringing together visual artists, scholars, composers, activists, writers, and others to explore what kinds of radical imaginaries can unfold in this moment of pandemic, racial reckoning, economic uncertainty, and environmental crisis.
We're interested in how we think about what is possible and how we can use our imaginations to build a better present and future. How we can retool and create better and more equitable models for living and working together. Donna, it's nice to see you again in this space.
It's been a while.
We've taken the summer away, so it's really a treat to be here. This is an exciting moment to share a great project. Will you tell us about our guest today?
Yes, so this is our second virtual program developed in collaboration with our institutional partner, the SETI Institute Artist in Residency Program. And today we're going to be talking about life. What is it? Can it be created on a computer or in a lab? And how do you conceptualize what life might be when you're imagining what and how you should look for it on other planets? We are joined today by Interspecifics, an independent artistic research bureau founded in Mexico City in 2013. And Interspecifics research focuses on the use of sound and artificial intelligence to explore patterns emerging from biosignals and the morphology of different living organisms as a potential form of non-human communication. Interspecifics are going to be talking about their new project, Codex Virtualis, in which they have used artificial intelligence to generate lifelike virtual organisms. And they have just presented this work at the ARs Electronica festival, and it develops out of a residency which came from a partnership between ARs Electronica and the SETI Institute Artist in Residency Program. We're also joined by SETI Program Director Bettina and Veronika Liebl, Director of European Cooperation at ARS Electronica and Managing Director of its Department of Festival, Prize and Exhibitions. And they're going to be talking about this very unique partnership. And finally, we are very lucky to be joined by SETI Institute research scientist, Rosalba Bonaccorsi, whose work includes astrobiology, environmental science, marine geochemistry, and biosedimentology. And her work focuses on exploring where life and its signatures for life are most successfully distributed, concentrated, preserved, and detected. So we have a really stellar group today, and I'm very excited to be able to share their thinking and work with our audience.
This is going to be fantastic. I'm looking forward to the takeaways. So links to our participants' bios will be posted in the chat. You can find them there. We also want to thank Nathan Zanon, our producer behind the scenes. And I want to be sure everybody's aware that you know the live captioning can be activated at the bottom of your screen as you're viewing the conversation today. We'll have the live captioning on our Facebook live stream as well. I'm going to disappear now. I'm going to leave this good conversation in Donna's hands, and our guests will talk for about 45 minutes. At that point, I'll return, and we will take questions from anybody who's listening either on Facebook or with us on the webinar, and field those questions with our group. Thank you, Donna. I'll see you soon.
Thank you, Kelly. I'll see you soon. So I'd like to welcome our guests into the room, into the Zoom room. If you could all turn your cameras back on and you unmute yourselves. Hello, everyone. Thank you so much for joining us today. I'm really excited about this conversation and to sort of dig into this question of what do we mean by life and how do you look for life elsewhere in the universe? And it's such a rich and exciting emerging field of inquiry. And I mean, we've got synthetic biologists in labs trying to figure out what's essential for life and trying to build cells from scratch, or, you know, taking existing molecules and trying to reassemble them into a living system. And at the same time, we've got, in the field of artificial life, researchers studying life and processes of evolution through digital experiments. And then meanwhile, we've got astrobiologists who are trying to understand what might be the signatures of life and where to look for them. So there's so much rich research and work going on, both in the scientific community and also, as we'll see, also in the artistic community. So we're going to start today's conversation with some brief 5-minute presentations by our guests talking about their work and research. And then we'll have a sort of 30-minute, 35-minute discussion, kind of digging more deeply into some of the really fascinating questions their work kind of provokes. And lastly, as Kelly said, we'll have Q&A at the end with you, our audience. So please post your comments and questions in the chat. So I'd like to start with Leslie, Interspecifics. Leslie is part of the International Collective and Research Studio Interspecifics. And Leslie, you just wrapped up a residency organized in association with the European Artificial Intelligence Lab, which kind of developed out of this very interesting partnership between ARs Electronica and the Institute. And you used AI to generate virtual lifelike organisms in a new project you're calling Codex Virtualis. And you just presented this work at the ARs Electronica Festival in Austria. So I'm wondering if you can start by sort of introducing us to the Interspecifics team and what you do, and then tell us about this really fascinating work on Codex Virtualis.
Sure, thank you so much. So I'm going to start here. Interspecifics is a transdisciplinary collective. I'm going to show you the members right now. So we have Carles Tardío, physicist and mathematician, Felipe Rebolledo, architect, designer and artist, Alfredo Lozano, machine learning engineer, Emanuel Anguiano, bioengineer, Paloma Lopez, researcher and communication scientist, Maro Pebo, artist and biologist, and Nicole Clitworth, artist and designer, and myself, sound artist and electronic designer. It's a very mixed group from different backgrounds, as you can see. And together we started developing different projects since 2013. Codex Virtualis is our latest endeavor and is the result of this residency with ARs Electronica and the SETI Institute. And we presented a first reference for these results in the festival a couple of weeks ago in ARs Electronica. So this is one of the first presentations. And we also designed a booklet, but I will say that this project is a long-term project. Usually it takes us around 2 to 3 years to fully uncover the different possibilities of the ideas we're working. So this is just the first delivery for us. The work is a research framework, and the installation is just one of the outputs. So you can expect a series of workshops coming out very soon. So Codex Virtualis is an exploration of algorithmic life, experimenting with algorithmic behaviors comparable to the mechanisms of life. Evolution not from competition, but from the collaboration and exchange of biological and algorithmic systems, such as the mixed training of organs with 2 qualitative different datasets that merge to lead into new visual samples that are analogous to the horizontal gene transfer and progressively fitting novel definitions of life. For this, we were inspired, of course, on the biological level. So we look for organisms that have special characteristics to uncover the challenges of extreme space environments, such as high levels of UV, ionization radiation, dryness. And we selected 4 organisms that gonna be the parents of our training process. And these 4 organisms have special abilities, special mechanisms, such as UV light resistance, radiation resistance, spore resistance to desiccation, symbiotic interspecific survival. So we use this data to train different data sets in order to acquire these specific characteristics. We are locating life as we know it within a larger picture of life as it could be, honoring, of course, computer scientist Chris Lanton. As an artificial life artistic research project, Codex Virtualis seeks to enhance our understandings of the genetic, metabolic, and behavioral processes of life by means of carefully articulating a display of scientific theories represented by a multitude of ecosystems of interconnected collaborative computer simulation strategies. As such, is concerned with a bottom-up scientific study of the fundamental principles of life. So our multi-layer computational ecosystem is formed by life form synthesis, representation, exploration, and generation, and what we call an ensemble of a display. So I'm gonna show you, just to be quick, because it's a lot of different processes, the 4 models we trained during the residency. So this is our 4 main systems: the Naro system, Lenia, Agua, and of course the model organisms, which are the extreme condition resilient organisms. And we use these 4 data sets in order to create novel virtual organisms. Just a quick reference of the kind of transfer of information that can emerge in between them. Here we have 4 different samples, same organisms, and the different recombinations of these organisms on the neural network with the help of the specific extremophiles. So you can see in the image how the extremophile, the model organisms, start transferring certain characteristics into the data sets. So I'm gonna be quick here, and I'm gonna show you a little video. These are some of our virtual organisms on the family taxonomical level. And here we are applying some computer vision techniques to observe the structures of the organism on a deeper level. And the 3-dimensional version of these virtual organisms with their own classification and names. So for us, all this started by asking ourselves about the possibility of finding on a morphological level some of the strategies of life that we could transfer as algorithms in our systems, and just the possibility of creating a system that is a collaboration of different parts and different algorithms. Also, the collaboration in between humans, algorithms, and virtual emerging entities. So I will stop here to leave a space for the other presenters.
Thank you, Leslie. It's just really fascinating. I just have one quick question, if you'll allow me.
Of course.
Can you clarify the data inputs that you're using as starting inputs to create these speculative life forms?
Of course. Let me go back a little bit so I can. So we trained 4 databases. Those 4 databases have different sources. So let me show you here again. So this is our main data sets. NARO is from the NARO Gene Bank. It's a public collection of 1,000 images. Lenia, it's a selection of solitons from a continuous cellular automata from the researcher Bert Chang. And AWA, it's a crowdsourced collection of images. The interesting part of AWA is that it came from different accounts on Instagram. Biologists that share their research in images around the world. So we contact them, we ask them to share their images to create this data set, and everyone was happy with the process, so they say yes, and we have, of course, all this data also available in an open source format. And the final data set is the model organisms, which are the extremophiles, and that is also something around 500 images, because it's much more difficult to find images from extremophiles as other kinds of bacteria, fungi, and different microorganisms that we are using for this exercise.
And can you tell our audience, just very briefly, if you could, what is a cellular automata? Because I think that's very interesting in regard to this question of artificial life.
Yeah, I would say that this idea of imitating life is as older as, medieval times with the automatas, which is a robotic system or a mechanical system that enacts some kind of behavior that is similar to a living being. So the cellular automata, it's an algorithm that is programmed to create behaviors that are similar to the growth, population, extrapolation of microorganisms. So there is several kinds of cellular automatas, and the more famous, of course, is the game of life, which is, I think, one of the first to be out on computer systems.
And so, they can create a kind of, to model a physical or biological system through a computer grid using specific algorithmic rules, right? That it can sort of imitate patterns of evolution. And
basically, they're very simple rules. They have like a number of neighbors, and depending on the motion of the neighbors around them, they can either grow or die inside a community. So it's quite interesting, because with very little information, they start recreating very complex behavior. And that's actually part of the beauty of this type of algorithms.
Yeah, well, I think there's a lot more to talk about here. So we're going to get into more of these questions in the discussion part. I'm interested in asking you more about the model for and how that relates to other models that are used to describe life, and whether we could think of these speculative life forms as alive in some way, or what would it take for them to be alive. But for now, let's turn to Rosalba. And Rosalba, thank you so much for joining us. And you are a research scientist at the SETI Institute, where you focus on astrobiology and environmental science and marine geochemistry. And you've been conducting various field experiments and research into the habitability of Mars analogs, when you've been traveling all over the world, right, to Mojave Desert and Australia, New Zealand, Death Valley, with this goal of looking for how we might identify signatures of life. So I'd love for now if you could share with us something about your work and research, and if there's anything in Interspecifics presentation that you'd like to respond to that seems relevant to what you're working on, then, you know, please feel free to do that as well.
Okay, thank you so much, Donna, for inviting me to these fantastic events. And yes, I would address, you know, possible convergence between my work and also what Interspecific has done is very interesting, just to start. And this is also why I'm presenting something that to give some feedback. So what I've been doing and how what I've been doing could fit what you guys are doing for the past and the present and future. So here we go. I start sharing my presentation. And okay, let's see. So we share screen. And, okay, can you see?
Yes.
Okay, but I cannot see it.
Oh, uh-oh. You need to see it.
Yes, where is it? Oh, okay, I can see now.
Oh, good.
So, it's going to be hilarious. Okay, here we go. Okay, why didn't start from scratch? Okay, guys, bear with me.
Preview, that's great.
Okay, this is just me, and I'm providing my email address so for being for contact by all interesting parties. Just I've been working at the SETI Institute that is pursuing the search for life, and everything from microbes to technosignatures, advanced cosmic civilization, and everything between. And we will be talking about today what is life, and one of the most intriguing topic that I do believe is around about this inquiry is life as we know it, but also what about us, life as we don't know it. And that's kind of, I'm presenting here like a famous Star Trek season 1 episode, I think that was 27, where they're dealing with ortas, which are silicon-based life. And the major problem of looking with life as we don't know it is just we don't know it because we don't know it, so we cannot see it. So this thought really troubles me sometimes at night, because related to how much we might have been missing. So how much we're just looking at the tip of an iceberg. And having said that, we can looking at places around the world on our planet where we have been looking for life at extremes as a most fundamental way, by looking at the habitability of extreme environments around the world. And these are just some sites, the red dots, where have been for over the past 16 years. And the yellow dots are just where I've not been yet, like the hardcore Antarctica or the Arctic. But most of my work has been focusing on subsurface life, or it's like Rio Tinto. This is our established SETI Institute, looking at life in rocks underground. And also it was being extended later on the surface life in the desert, because they are more easy to access depending where they are, with more focus on that valley and Mojave Desert, because it's in our backyard. Me being based in the north to central California boundaries, more easy to go to these places for running long-term investigation. So looking at, not just in space, but also in time of how life would evolve. So when you look at life and also is very important, particularly in extreme environments. It's very important trying to not to be bothered by contamination, because life in extreme environment is very sparse. And when we're looking at collecting this life and sampling these lives, for instance, doing drilling simulations, like the one we're doing in Atacama Desert, where life is very rare, scattered, it's just also trying to mitigate contamination. This is an important thing. So part of my work was also cleaning and sterilizing and tracking contamination that might be introduced by humans doing these tests. And this involves, of course, looking at proof of contaminants like biomarkers like ATP, adenosine triphosphate, that have been using a lot because it's very simple molecules that could be looked at in situ, so instantaneously, and it helps very much in doing this type of work. So other places around the world we have been going to, it's one of the most interesting I've been was the Indian Tibetan plateau, doing a spaceward-bound expedition. So a spaceward-bound expedition were first conceived at NASA Ames by Chris McKay, by going into the field worldwide and involving communities there, but particularly also to train science and educators by involving them in doing active science. So, so we were everywhere, for the Mojave Desert to the Atacama, starting 2006 to nowadays. This spaceward-bound expedition has been evolving to involve no-profit activities and no-profit communities and institutions, and also here an example of the cool stuff we could do by looking at extreme elevation desert, but also involving nomadic communities, in particular those children. So this was an example of the most extreme poverty in the world. I don't think there could be even more extreme poverty. But yet, kids really love to be given the chance to experimenting with their surroundings, as very poignant experience. And this is what an example of what you just mentioned about my work. Like, I go places and I still use the ATP luminometry. That's a very simple tool to look at the total biomass in all these environments, like very rich in salt or very poor in nutrients or clays. And this is kind of a nice example of one of these high elevation lakes where you have a brines. It's almost like you can walk on these brines. And these brines contain a lot of biomass and a lot of microbes. And it's very interesting, like salt, it could be viable environment for life too, contrary to what was thought some probably decades ago, that life couldn't survive in salt. This is about Mojave Desert, just showing where it's located in the heart of Central California. And that valley here is a part of where the asterisk, the highest stars, is a part of the Mojave Desert. It's just the extreme of the Mojave Desert, because the temperature can reach up to 54 Celsius degree, actually, as we probably know for the news of the highest and highest temperature seen in that valley. Research in that valley also, and Mojave Desert started in relationship with the spaceward bound. So we went over there for many, many years, bringing this crew of teachers. We did mini rover testing, study of biological desert crust, and the Mojave and our deserts all over the world are an example of a Mars first billion years water cycle, because the water of Mars in that time was probably present there, but was very, very sparse and was dying out. As Mars was dying out, also the water cycle was shutting down. So this is why we're looking into the desert. This is a nice example of desert biomass. And just by adding water, the magic ingredient can just boom, like creating instant life. This is not the trick, it's not being accelerated. It's just real time. And you can see before adding water, only a trained eye could look and see that there was real life, right? Otherwise, you would, just kind of dirt, but it was not really dirt. And moss is not really even considered extreme, like extreme microbes, but you can see in the desert, mosses behave like an extremophile. So this is another place, the UBB volcanics, that is located in the northern part of that valley. We've been, working for the past 10 years by looking also at not only microbes, but particularly the water cycle. So I've been doing consistently monitoring, because before looking at microbes and biomass and extremes, we really need to characterize the extreme parameters like temperature, moisture, light, and other things. And I've been looking at extreme ephemeral lives, like very fascinating, like this instant life where microbes can just pop up to life after just you add water, in this case by rainfall. Other part of my work was back to the ocean, right? I started with the ocean as a marine biologist many years ago, and I was very fascinated by the search for life on Enceladus by way of missions that will go through plumes. Plumes are ejected by Enceladus through the South Pole, and they're thought to be the product of something that happened beneath the ice sheet over there. And so missions has been planned for going back to Enceladus after Cassini and sample possible life in plumes. But here's the trick. As you see, we have a lot of analogs for Mars, but we don't really have analogs for Enceladus on our planet. So we have to do that in the lab. So over the past 4 years, I've been following up with, been engaged actually at the very beginning with the research for simulating an Enceladus plume in a lab. So we do this at the NASA Ames. The facility is called NASA Ames Vertical Gun Facility, that you can see here, has a chamber where we can simulate space conditions, and the plume is generated by oblique impact by using this gun to shot projectiles and produce ejecta plumes of icy grains. And of course, over the years, we have been able to refining this concept and training the plume to behave nicely and be measurable in a way we can really look at the speed, ice grain dimensions, and we can use the plume then for running a lot of experiments for collecting the plumes at a speed about 2 kilometers per second. So we need the impact, we need to simulate that, and then we can look at the, we already tried to look at the microbe survival in this ice by using ocean analogs. And that's part of the work I've been engaged. Just a couple of words about the feedback between education and science we have been doing during these expeditions. So we have been working with the local community by, for instance, in Australia in 2011, we do spacesuit trials. And of course, that's by serendipity happened that we were spotted by Aborigines kids. They came and really enjoyed what we were doing. So it was really kumbaya moment where the future met the ancient past. And that was really, I think, the most important things that have been done after Space War India. So this is how I see science and the role of scientists. I would see the worldwide engagement of humanity. So if science is for build social connection and share the meaning of us being human, also science for inspiring people. And it's important, look at like synergies between art and science can change people all over the world. And the take-home messages all over the world dealing with very affluent people and very poor people. But both of these extremes in the societal range are really engageable by science. So we're all equal as a humanity to that extent. Other works have been done on organizing Mars Fest event in that valley by running family-friendly expositions, science talk, and scientist-led field trips. And I started this program by working with the the National Park Service for 2012 to 2019. Program is still alive, but under a different agenda. And last but not least, work with the RAU, the research experience for undergraduate program, done by SETI to engage undergraduates and funded by National Science Foundation. And we've been using all this delay of all experience all over the world with teachers for running a real field experience with students for a week or so, both at Lassen National Park, the Hat Creek Radio Observatory, and in the California Central Coast. And thank you so much for your time. I think I'm exceeded, but thank you so much for being here.
Oh, thank you, Rosalba. It's just incredible, fascinating work. And thank you for sharing it all. And, you know, you asked this super fascinating question about, you know, how will we know what life is when we see it? And how are we going to be able to distinguish a living organism from something else? And that's something I hope we can really dig into a bit more when we all have a bit more of a discussion, cross conversation. But now I just want to invite Bettina and Veronika to join us and to tell us a little bit about this like very unique partnership that came about between the SETI Institute and ARs Electronica to support Interspecifics and this incredible research, and why your institutions are particularly interested in this sort of topic of artificial synthetic life and artificial intelligence, and what's sort of driving that.
Thank you so much. Bettina, do you want to go ahead or should I?
Yeah, I'll go ahead once I unmuted. I was just so delighted to work with ARs Electronica on this project because we have such a great alignment in our mandate. Both of us are interested in the intersection of art science and technology, and also with the purpose of asking bigger questions. For ARs Electronica specifically is looking more outside the societal impact of that art science and technology intersection. For us here at the SETI Institute, our focus of the Artist in Residence program is also to look at the big philosophical questions that are posed by our scientists. And I love what Rosalba says, like, how can we understand life that we don't know? How do we even think about it? And I think our scientists are doing a phenomenal job at imagining the unimaginable, but artists are specifically trained at imagining things and coming in with unusual questions and new perspectives to generate new insights. The SETI Institute is very interdisciplinary already by its sheer virtue of having astrobiology and astronomy and machine learning, and we have a lot of different foci, like what is intelligence, what is the evolution of intelligence and communication. And I like to match our artists with these research focus points. And of course, Interspecifics was just perfect. I mean, it could not have been better. I was so delighted, and I'm just stunned with what they've come up with. And Veronika, I'll let you talk about the focus of Future Lab and ARs Electronica.
Thank you so much, Bettina, and I can actually only echo your word. It is so outstanding to work with Interspecifics, and I actually had the pleasure to see also the first iteration of Codex Virtualis just 2 weeks ago at ARs Electronica Festival in Linz, and it was really stunning, and it also received so outstanding feedback by audiences. So congratulations, actually, mostly really to Interspecifics. Yeah, and also to come back to your question, well, I mean, exactly that's the reason why we're doing this. We really love these encounters also between art and science, or more precisely also between artists and scientists, and also this clash of disciplines to emerge also curiosity from and understanding also for audiences. And of course, that's also one of the core missions of ARs Electronica since the very, very beginning, 42 years ago already, to bring artists and scientists together and build this common understanding, and also to build a mutual curiosity. And it was absolutely brilliant to find with SETI another organization who shares also this excitement, and that's definitely not something which is given, also a willingness to have an open-ended artistic research process. I guess our both interest was really also in finding a residency who's addressing these fundamental questions, as you said, Bettina, also going beyond the superficial and questions about what it really means to do the science, but also what it means to understand the universe in general, questions that also really launch reflection in all of us together. And that's what at least we've been looking for. And with Interspecifics, we found really a collective addressing the ultimate questions of our life, who we are, where we come from, our evolution, also in relation, of course, to autonomous systems. And one of the big factors I also have to mention, of course, in the scope of this specific residency was their approach working with artificial intelligence, reflecting probably also the biggest of all questions currently: what is AI and what do we actually know about human intelligence? How intelligent can artificial intelligence be in comparison? And in our daily work, at least here in Linz at the Ars Electronica Center in our museum, we experience plenty of room of speculations, also questions on these questions, like how many of our tasks do we actually want to outsource to machines, and will AI ultimately take over? And this is exactly so important for us because we try to give these questions and this discussion back to audiences. And we see also that they are driven by curiosity on the one side, but also insecurity in some time. And we see it also as a mission of Ars Electronica to shed some light on this and create a basic understanding. And that's why we were in particular intrigued also by Interspecifics' approach on creating open knowledge, on also creating open source materials. So thanks again for this really, really fantastic collaboration.
Thank you.
And I would just like to add a small thing. Veronika said there was like a clash of disciplines between art and science. I also think there's a lot of overlap between art and science, especially in the area of research and the kind of research questions our artists and our scientists are pursuing and the way they're approaching these research questions. So I think art and science have a lot more in common than is generally assumed, which is why these residencies work so well and are so interesting.
Yeah, it's a really inspiring model, and congratulations to all of you. So I'd love for us now to dig down into some of the questions that this work raises. And I mean, I have questions, but I'm sure you both all have questions. So let's all get engaged, and please, feel free to weigh in. But I mean, I think my point of departure is kind of going back to this idea of, how will we know what is life when we see it? And how do we distinguish a living organism from something else? Like, why is an anteater considered life, but a hurricane isn't? And I know NASA has a definition, which is life is a self-sustaining chemical description capable of Darwinian evolution, meaning it's got heritable variation leading to reproductive success. But I also realize that there's quite a bit of pushback against that definition as being too limiting. And I'm wondering, both in, your work, Leslie, with Interspecifics and Rosalba, how are you conceptualizing what life might be, when you're either creating models to generate new life forms, or when you're actually going to look for it? What is it that what's the basis? What was your concept for life when you're doing that?
Well, it's this kind of very profound question. So I will say a definition of Ions Earth from Penny Boston when she came to give a wonderful talk at the Mars Fest to this big audience, like, what is life, right? And Penny has been very knowledgeable scientist with very broad experience, really quintessential and broad. I really bow to her, like she's my, goddess in terms of example. So she operationally defined life, everything from, a plasma cloud a microbe. So she included a plasma cloud, which is probably not a very chemical entities, but more physical. And so this might connect to, I recall there was some, probably the Russians, the Academy, Russian Academy of Science, maybe 20 or 25 years ago. They had scientists talking about like a clay phyllosilicate system might have some resemblance with the operational definition of life, like, you know, how the fact they can reproduce, they can grow, they can move, you know, in space, they can interact in interesting manners, how the mineral life could be, you know, something that we could talk about it, right? You know, sometimes it's taboo, say, oh, are the rock alive? Or, but yeah, there could be kind of expanding our horizon beyond, even more to go and bulk to a plasma cloud, you know, let's start talking about that kind of life, if any. Of course, we need to be, as a scientist, be able to test that. But just so you know that I've been talking by people from Russian scientists. So it's not really kind of woo-woo question to be asked. From my perspective, sometimes I will be pessimistic about our possibility to see, not because I'm a pessimistic, but because I see, like, even when we look and deal with the life as we know it, like simple biomass, right? And we go to a place like Atacama, but even in our backyard, I realized that what doesn't matter at the very beginning is like our capability to see something is already there. And any instrumentation, particularly when you look at microbes, normal mundane microbes, right? Simple guys, not on a petri dish where we can monitor and their growth and clearly see them with a microscope, but microbes that act and live in minerals or in media. The problem is the detection. So detection of sometimes alive using like ATP luminometer or biomarkers detection tools, they deal with the physics. So we look at, you know, how an antibody fluorescent molecule could bind to a molecule, a biomolecule from a microbes, and then looking at, you know, UE, infrared or flash lights emissions, and then we detect that. So these are physical detectors of some chemistry associated with life going on. So because physics and chemistry are very much involved with the mineral environment, sometimes they do interfere with our capability to see something. So I have a kind of problem of signal to noise ratio, where the signal is the tiny life we see over there, and the noise is done by all these interactions that produce noise. So we might not be seeing them, so we might have a false negative. And for instance, you know, we look 1 year, I remember we had a project we had by looking at DNA in clays. And I used this sample and said, well, great, these guys are going to your master in detection of DNA and looking at more refined, you know, metagenomic studies. So here we go, a sample of Mojave Desert soils where we know that we have up to, you know, 500 millions of microbes per gram. So I hope that there are plants there, you clearly know that there must be a lot of DNA over there, just as an example, right? But there was a clay sample, like one of the most, you know, intriguing and challenging sample for looking at microbes. You know what the result from the lab was? There was no DNA in the sample, just because the clay at nanophase level interfered with the capability of whatever they were using for extracting DNA. Sometimes you cannot even extract, or you can extract and then you don't see it unless you apply particular protocols. And I saw this over again. So sometimes I'm really very concerned about the basics. It doesn't matter how much sophisticated is the state-of-the-art equipment we're using, right? It's more sensitive. The fundamental problem that every state-of-the-art light detection instrument would bump into it would be this false negative issue, even with simple things. So my input in this conversation.
No, that's fascinating. And so, Leslie, what's your kind of perspective on?
Well, I first will love everything that Rosanna was talking about, and I would like to point something that for us is super important. This thing of tools, equipment to analyze processes outside, and the relation they have on the construction of the reality as we perceive it. So if, as Rosanna says, we have a bunch of false negatives, all over science, then we have a lot of questions of what is actually, for example, life. And one of the main tools we have to do that is our perceptive senses. So we are delimited by our senses and the idea of constructing all this reality. And we start from there. We start like asking about life beyond what we can perceive and trying to expand this perception by, the senses of the others. That's why it's so important for organisms and plants and everything that has other abilities to perceive light, sound, information, and transport that information on a different level. And we try to interface with those organisms. So essentially, we like to see us as some sort of living sensors, sensors that are capable of evolving over time. But we have in our own system everything we need to grasp that reality. But at the same time, that capabilities are being delimited by so many things, logics and cognition, language, human perception in general. And we try to expand that from other cultural approaches. For example, the Latin American cultures in general, the cosmovisions of our ancestors, they say that almost everything has the capacity to experience the universe. And in that sense, almost everything has some sort of intelligence and some sort of way of expression. And that for us is a big, big inspiration. So we try to combine today's technology with these philosophies in order to create art that in some way, enhances this idea of the agency in matter. But at the same time, that's something coming on from, the new materialism philosophy and different kinds of object-oriented ontologies and different kinds of cultural developments that talk about all these processes. So in that sense, a rock is something that is transforming over time, that is unstable, but not in our own timeline. We need to speed up in order to see that transformation. And when you speed up the velocity, then you see that these things are actually very similar to the behavior of some kind of bacteria. Or when you slow down the speed of a bacteria, then you see another thing. So it's a game of perception in many senses. It's a thing of speeding things up, slowing things down, changing devices and trying to, be with an open mind. Of course, we can make crazy questions. Nobody's like delimited on us on that. Sometimes for an artist, it's actually quite, respectable to make those sorts of questions. But we like to make these questions also with scientists. This is important because at the end, it's not about, the technique you're using. It's about the knowledge we're looking for. So in that sense, I think we are in the same road. We are looking for knowledge. We're looking to answer these questions. And ultimate, we are looking to ways that can describe life in a way that makes life in this planet a little bit more equal, a little bit more, equilibrated in that sense, but also open our minds to experience other ways of lives and maybe learn from that different ways for us as humans.
Do you think that the speculative life forms that you're creating would be considered life now,
well,
future?
I don't know. Right now, our question is not that much if this is considered life as it is, but we are asking more questions around what can these morphological, aspects that emerges in life can tell us about these specific organisms, and how can we learn from these, inner workings of the algorithm of life. That's an idea we have, like it's possible to reproduce this algorithm. If it's possible, what kind of technology do we need to do that? And maybe in the future you can say this is life, but for us it's more like start habitating the technosphere space with different types of possibilities. Like go beyond, the consumer technologies, the military technologies. So when you have artists creating these kind of things, we are putting these possibilities out there in the same space. So it's more in that direction that trying to affirm that this is life. It's more an open question for us.
Yeah, I understand. So Bettina and Veronika, I just want to invite you in and see if you have responses and thoughts about this as well before we can open up the Q&A a little bit.
I love what you said about the algorithm of life as a quest to understand where life originates in your Codex Virtualis. And I think you're calling this section Codex Virtualis Genesis, just specifically to focus on that question of what life is and where it comes from. I think those questions are related, and that sort of ontological question about life, I think also connects to, in the humanities, how we understand what life is, that it's much more networked. It's no more this hierarchy of there are lower life forms and there are higher life forms, but that is all like this sort of rhizomatic approach that also includes, like if you're looking at thinking of actor network theory, that everything has agency, including non-biological actors. And I think in the sciences, we're inching toward that perspective as well, that maybe our understanding of life is too biased toward biology. And I remember Natalie Cabral from our head of the Sagan Center, she in the talk said that even maybe the universe itself has consciousness, is a conscious entity. If you're looking at quantum theory and entanglement, who's to say that the whole universe isn't conscious? So going, I find that these kind of two streams of thinking about what life is kind of are crossing, and the results are really fascinating.
Yeah, that's so interesting. Like, we don't have to just get fixated on this idea of the definition of life should involve an individual organism. It can be much more expansive, and I think that's such a great point. Veronika, do you have any thoughts?
I mean, I found just the discussion now so interesting, as it also reflects very, very much our current discussions with audiences here in the museum and also at the festival, because it is also reflecting again the topic of agency at the end, and also the future which we're looking towards, to which will be a cooperative one between also algorithms and life, between humans and machines. So this hybridity is definitely something where we will actually face a lot of discussions, or the urge also to discuss not only how that will look like, but how it should look. It will be really based on our decisions, our understanding also of what we actually envision for our society to happen. And this is exactly also a question how then such systems and by whom such systems should be developed, deployed, used, and monitored. So it goes very much also in ethical discussions, which is definitely something, of core interest for our discussions here.
Well, thank you. So let's bring Kelly back on and see if we have some questions from the audience. This conversation could go on and on, and I would love it too, but I'm just being mindful of time.
This is a dinner conversation. There's many, many things. This moment we're living in and what sort of has captured all of us and keeping us at home. I think of this tiny, tiny virus and what kind of intelligence lives within that. We do not have questions, but we probably have raised many questions with people listening today. So I just want to thank you all. This was incredibly fascinating and really feels, this world of AI living in Silicon Valley, we've sort of watched it be born and evolve over the last decades, and where it's going and how this idea, Veronika, of this sort of relationship between algorithms and sort of biological creatures is, it's fascinating. And I think this is what we're going to have to start figuring out. Thank you. Donna,
Thank you.
do you have a closing question?
Well, I did wonder if Leslie, you could tell us, what's the next phase of the project?
Sure. Right now we have available a booklet we designed to present the first stage of the research process. So I will put the link here if anyone wants to download it. It's free in digital format. And we are continuing to the second stage, which is Codex Virtualis Habitat. And in this stage, we're trying to make the organisms to actually interact with the specific conditions outside, like these extreme conditions we were talking about. We want to see if our model is capable to create, any reactions, because it's a whole model with the DNA we are using. So each organism has 50% of the parent DNA and 50% of the generative DNA. So in this testing model, we want to see how much the parent model is like transferring the abilities to survive these extreme conditions we're going to try to recreate in a virtual sense. And that is going to be exhibited on the ARs Laboratory in Berlin on the spring. So we're going to be working on that. We're going to start doing some workshops around the first stages of the process, how you can use our neural network data sets and start like just seeing these organisms pop up on the screen, because they're really beautiful actually.
Yes.
And we have some tools for them, like you can organize taxonomical families. So I think maybe for biologists, it could be a fun thing to do on the spare time, just to see what's going on the neural network.
Well, Leslie, what you say is kind of like really match my hardcore.
Yeah, I know.
Interest about extreme environments, and I always wonder how the music and the songs of those microbes in Denmark from -16 or 9 in winter to plus 60 something 67 or 70 Celsius degree in summer would sound like.
Yeah, sure.
So that would be kind of would be super cool. Like,
that's the thing we love to do actually. So we for sure gonna be in contact. We can later have another session to talk about this idea you have, of course. And yeah, we love to do sound of bacteria going into different stuff. So for example, for this piece, we try to simulate something close to a community. Like when you are in the rainforest has their own sonority because of trees, crickets, all the living matter in there creates an acoustic symphony. So we try to follow these rules in order to create the sound for our own installation. So we use different data triggers from the neural network, and we ended up having a piece that is slowly morphing over time, but it has a signature. So it was like sort of like an echo virtual signature of the matter we were creating. So yeah, it's a subject we love to approach.
Well, we're looking forward to seeing and hearing more about the new phases of the project. I want to thank Rosalba, I want to thank Leslie and Interspecifics, Bettina, Veronika, and our audience for joining us today. Thank you for such a fantastic conversation and for sharing all your amazing work and thinking. We are planning some new Scratchspace episodes for the fall winter season, but so please check out the Montabo website for any updates. And Kelly, do you want to tell us about what we have coming up in terms of on-site programming in
Absolutely.
September, October?
I just want to join you in thanking everybody, and our partnership with SETI, brings us these conversations. Bettina, thank you so much. Rosalba, thank you for joining us today. Leslie, it's a treat to meet you. We'd like to meet the whole team. And Veronika, it's a treat to have ARs Electronica and you here with us today. So coming up at Montalvo, we have our public park and people want to be out. I think Veronika said she could see this in people as they entered the festival, and we've had the same experience. Next week, on Thursday the 30th, we will be celebrating with artist Hector Dionisio Mendoza, the Latinx Heritage Month here in California. We have many, many local performers who will be participating and performing on the veranda of our historic villa. We'll be doing art making, workshops, have a special conversation with Dionisio, and he is going to share the gift of a scholarship for a young arts practitioner to come and spend time with us at Montalvo. So we're looking forward to that. And in October, on the 21st, we will be developing a new work of theater. It's actually a historic piece that's being created by Andrew Sato and collaborating director Miranda Heyman out of Boston. Together they'll be in residency, working with 4 actors, and they will be doing a public reading of a work in development. It's a historic story about American POWs in Japan and radio broadcasts that were created to instigate racial division within the country. So we're looking forward to that work. It's entitled currently Fire in Heaven. Hope you'll join us. Can't wait for more Scratch Space. I actually have come to love this space for conversations, and I appreciate you all being here today. Thank you, Donna.
Thank you, everyone.